Welcome friends to another interesting video. We are talking about some aspects of cell division in which we had a previous discussion regarding. So in that we came to know that cell division has got three different phases. That is the interphase, karyokinesis and cytokinesis. And the next video we had a brief discussion of. So in continuation, we have to talk about karyokinesis, that is the nuclear division, which can happen either through mitosis or meiosis. So before which I would like to throw some lights or have discussion of exactly what a nucleus is, not the structural aspect, but what nucleus contain, that is the DNA, chromosome, etc. etc. Very basic aspect, so that we have a clarity to understand events that are happening in karyokinesis, that is in mitosis or meiosis. So in this regard friends, I would like to say that I have as a very similar discussion in a video entitled A Journey from Gene to Chromosome couple of years before. Though I have that video, the related video done by me, but still I would like to make this video to have a continuity, right? So even you can watch this and you can watch that and get information wherever extra information is found. Friends, with this, let's get into the discussion. Say you have a cell. In short, the cell has got a central part which is called as a nucleus. So let's get this nucleus out. The nucleus, okay, it is a double membrane structure, etc. etc. So as I said, I am not getting into the structural aspect of this. This nucleus has got the genetic material which is called as DNA. Okay, which is called as DNA. DNA is considered as genetic material because it contains the information regarding cell. What information? Our cells are basically made up of protein, right? So proteins are in turn made up of amino acid. So the difference between two proteins is the sequence of amino acids, length of amino acid chain. So all those information with respect to a protein is there in the DNA. Hence DNA is considered as a genetic material. It is considered as a genetic material. Hope you get this point. Now, you could see that this DNA is a double-stranded helical structure. Friends, here I would like to say that this double-stranded helical structure is only with respect to eukaryote. In prokaryote, it is going to be double-stranded circular structure. Even in eukaryote, the mitochondria and chloroplast, which are considered as a semi-autonomous organelle, will have a double-stranded circular structure. So, okay, so get these points into the mind. See friends, you know it has got two strands. You could see that. Okay, so if I'm going to unwind this strand, I can label it as 5 prime, 3 prime, 3 prime, 5 prime. So this 5 and 3 has got a significance why we call it as 5 prime and 3 prime. And friends, as a protein is made up of amino acids, as a carbohydrate is made up of sugars, that is monosaccharides, as this building is made up of bricks, okay, this DNA is made up of something called as nucleotide, N1 till NN. Again, this is going to be NN till N1. This is what is NN because the length of the DNA, which is nothing but the number of nucleotide, varies from DNA to DNA. One can be 1000, one can be 10,000, right? So in short, this DNA is made up of nucleotide. Okay, friends, this nucleotide has got three components. One is a sugar component, which is a pentose. That is, here it is going to be a ribose. The other one is going to be nitrogenous base, which can be either a purine or a pyrimidine. Purine can be an adenine, guanine. Pyrimidine can be cytosine, thymine. Though there is an additional uracil here, but uracil doesn't get into the DNA nucleotide, whereas uracil is going to be the part of RNA nucleotide. And you have phosphoric acid. So this is going to be the structure of a nucleotide. And friends, you could see these two strands are anti-parallel. What do you mean by anti-parallel? Both are parallel, but they run in opposite orientation. That is N1 to NN, NN to N1. That is, you have a 215 centimeter scale, 0 to 15, 15 to 0, right? So this is basically a structure of DNA, okay, which we consider as a genetic material, because this DNA, in the molecular genetic chapter, you will read that this DNA is going to contain the information regarding different protein, okay, without which the protein cannot be made, and without protein life is not possible, right? Fine. Next, friends, this DNA, in case of human, if you try to unwind and measure it, it is going to mention about a 2 centimeter. The question here is, how this 2 centimeter DNA is getting inside a microscopic cell, that too inside a nucleus, which means that the DNA has to undergo as compaction. Okay, Tamil surungarudhu solvanga. It has to undergo compaction. I always say during my classes, I give you a big bed sheet and ask you to take home, put it inside a bag. Okay, so what do? You try to compact, try to fold it, put it inside a bag. Likewise, this DNA undergo a compaction, okay, thereby it become a structure called as, that is, this DNA become a structure called as chromosome. Okay, chromosome. This is going to be DNA. And this chromosome has got a central part which is called as centromere, and you have this as a chromatid, CHROMATID, right? And friends, your book will say you that based on the position of this centromere, you are going to classify this chromosome into metacentric, submetacentric, acrocentric, telocentric, where if centromere is going to be exactly at the center, metacentric, near the center, submetacentric, end at the fag end, telocentric, near the end, acrocentric, right? So this is the centromere, and you have a primary construction. Sometimes there can also be a secondary construction somewhere here. All those things are going to be the part of this chromosome, right? Now, friends, this compaction of DNA into chromosome doesn't happen in a single step. What happens is, actually the DNA become a thread-like structure which is called as chromatin, and chromatin wounds around a protein called as histone, like this. So you have a histone, okay. What happens, you could see here that chromatin wounds around histone like this. Then this structure, DNA becomes this, takes this form, that is, it is called as bead and string appearance, and from this structure we end up with a further compact structure called as chromosome, right? Friends, here two things to be noted. This is a chromatin, as I have mentioned here. This is chromatin, and here you could see there is a histone at the center surrounded by a chromatin. The red one is histone surrounded by a chromatin. You call this as a nucleosome. So I can say you have nucleosome 1, nucleosome 2, nucleosome 3 and nucleosome 4. Things keep going on. And each nucleosome is connected by a chromatin again, which is called as a linker chromatin. Okay, linker chromatin. Hope you get this point. Okay, so this is one thing. And second thing, one important thing, that DNA is made up of nucleotide. We have seen a couple of minutes before, and nucleotide is made up of phosphoric acid, which provides acidity to the the DNA or the nucleus. So which means that the histone histone is basically protein. What is protein? Protein is made up of amino acids. So this histone is made up of some basic amino acids, okay, that is arginine and lysine. So this is going to neutralize the acidity provided by the DNA, phosphoric acid present in the nucleotide of DNA. right? So this is the basic structure. So we took a nucleus, DNA, and we had come to the compact structure called as chromosome, right? So friends, in another discussion we will talk about the various chromatids in the chromosome. There is sister chromatid, non-sister chromatid, homologous chromosome, heterologous chromosome, okay, in a separate video before we further get into the discussion on karyokinesis, that is beginning with mitosis, right? So I'll be with that video soon. Thanks for watching.